Hydrolysis of proteins. The learning intention is you should learn how proteins are broken down to amino acids by the process of hydrolysis. Understand the digestion of dietary proteins by enzymes, be able to draw the products of hydrolysis for a given protein. Success criteria: understand and be able to draw the products of hydrolysis when given a polypeptide, state the reactants required for hydrolysis of proteins, explain how hydrolysis is performed in the lab. Okay, so let's check your previous knowledge before we continue. I would like you to do the following on a piece of paper before you continue to the next slide and see the answers. I would like you to join 3 alanine amino acids together. I would then like you to circle the amide link on your diagram. I would also like you to state what the amide link is also known as. And can you give me the general chemical name for proteins? Have a try at it just now before you move on to the next page. So let's look at the answers for this. So you have to join 3 alanine molecules together. So like that, we just draw one out. The carbonyl group points up like so. What would have happened, I'll draw the functional groups back in just so as I can explain how this works. So what happens here is we've got the 2 alanine molecules and we have to draw the loop around the H and the OH, and those 2 parts are going to come together to make water. So I'm going to put the water down there, and now I'm going to rub out the H and the OH and simply just join that line up together, just like so. And then I would be doing that same thing again at the other side. So I'm going to rub the OH out, move the carbonyl up a little bit, and repeat that pattern that you see here. The N, the H, C, a H, CH3, another C, and because this is the last one, I'm going to put the carboxyl group back on just like so. Now you have to circle the amide link. Hopefully you managed to do that. That part there that I've circled is the amide link. What is the amide link also known as? It's also known as the peptide link. And what is the general chemical name given to proteins? Well, proteins are known as polypeptides. Hydrolysis. Hydrolysis is the opposite of what you did making the polypeptide, polyamide, or protein. Hydro means water, lyse means to cut or to break up. So lysis breaks up the protein. It forms 2 peptides when you take a dipeptide. So here we have what we would call a dipeptide. Okay, dipeptide is just 2 peptides joined together, and polypeptides are your proteins. They're many peptides joined together. But to keep the example simple, we've got a dipeptide here, and water is forced into the peptide bond, and as you can see on the right hand side of the arrow, that causes the amino acids to separate into the 2 amino acids. The smiley faces have just been placed there in order to show the functional groups only, to keep the diagram as simple as possible. This is of course a simplification, as a protein has many peptide bonds and lots of water is being used to break lots of peptide bonds throughout the whole of the polypeptide. What I would like you to do is, I would like you to draw out the process involved describing the hydrolysis of this molecule that you've got here. So on paper right now, could you pause this video or this PowerPoint, and can you draw that molecule out And can you describe the process, showing where the water would be entered and showing the products of hydrolysis that would be formed? There is no requirement to name the amino acids. However, if you could do, that would be great. So here's the answer. So there's the molecule there, and the water is forced into this section, the section shown, and it breaks up into 3 amino acids. One amino acid on the left is different to the other 2 amino acids on the right. Now, how does hydrolysis actually work? Well, hydrolysis is done in the lab using equipment that's shown. In the lab, a protein can be hydrolyzed back into its constituent amino acids by refluxing with concentrated hydrochloric acid for several hours. When that's taken place, you can then test for the amino acids that you have in your round bottom flask. The amino acids can be identified by the use of paper or thin layer chromatography. A piece of chromatography paper is spotted with some amino acids suspected as being present and also with the hydrolyzed protein. By comparing the position of the spots of the known amino acids with that of the hydrolyzed protein, the amino acids in the protein can be identified. Okay, so here we have hydrolyzed fruit juice is this part here, which I'll just highlight by doing a little dot above it. And we think that there might be aspartic acid in it, there might be leucine, and there might be lysine. So what would happen is you would put this in the paper chromatography or thin layer chromatography. Those amino acids would move up the paper, right? And perhaps you would have a band here, and a band here, and a band here, right? And you would then have the amino acids that are found in the hydrolyzed fruit juice, and you would look to see if there is any bands in the same line, right? In the horizontal plane of the paper. And as you can see then, in this example that I've drawn, this hydrolyzed fruit juice actually contains lysine. It contains lysine. Just a point to note that amino acids are not coloured, and so therefore you would have to treat the paper or the thin layer chromatography silica plate with something to expose the bands, allowing you to see the bands. Okay, so that is everything that you need to know about proteins and hydrolysis.